So let's talk about reproductive physiology. And the principal function of reproductive physiology is to increase the genetic diversity within offsprings. The way in which takes place is through the process of gametogenesis. Gametogenesis is going to be producing gametes through the process of meiosis, in which we're going to generate from the ovaries oocytes and from the testes sperm from the primary oocytes within the ovaries and the spermatogenic cells within the testes. The processes that are going to be producing the gametes are going to be regulated throughout reproductive lifespan with decreasing effectiveness as the individual ages for both males as well as females. So remember that what we're producing here are going to be sperm from the males and the ovulate from the females. These are not eggs, regardless of what might be stipulated within textbooks or how you might discuss them. These are follicles and ovulates. The oocyte within the ovulate will be the reproductive cell, whereas the ovulate itself is going to form the reproductive terminus for the female. This is also going to be the structure that will go about implanting following fertilization. So why do we need the gametogenic process? The gametogenic process is going to ensure that we get cells that are genetically diverse from each other through the processes of crossing over, as explained by Mendel's laws of genetics, including the law of independent assortment and the law of segregation of alleles, as well as cells that have one half the total complement of genetic material at the end of the meiotic process. We need this half complement of materials so that when the two gametes fuse during the fertilization processes, we have a full complement of genetic material within the zygote. This ensures that we have viable offsprings stemming from the reproductive responses. So let's take a look at Mendel's laws and how it's going to relate to our reproductive physiology. And then we'll take a look at a couple of examples. There are more examples within the study packet for you to use. So the laws of independent assortment tell me that chromosomes that result in fertilization are randomly sorted out of all of the possible combinations within the paternal or maternal chromosomes. So what the genotype for the zygote will be is randomly selected based off of all of the possible combinations. And so we're able to determine a probable and a possible outcome, statistically speaking, based off of what parental genotypes and phenotypes might be. Whereas the segregation of alleles tell me that during the process of forming the gametes, with the exception of alleles that are too close to separate from each other, all alleles will separate from each other during the processes of meiotic division. Now, what we found subsequent to Mendel's laws of genetic diversity is that there are alleles and genes that are linked because they're so close to each other that they cannot cross over and separate away from each other, which means that we usually will see these distinct genotypes in sequence with each other as opposed to being completely segregated out. Now, a lot of what Mendel's laws are giving us is it's giving us a statistical asset and assessment for what is the probable and the possible offspring outcome following reproduction. One of the ways in which we can examine this is through the expression of what is there, what the genetic material happens to be, in which we have two distinct terms. And the two distinct terms that we're looking at is phenotype, what is being expressed, and genotype, what is the actual genetic code. And this is where we have to be very careful because just because I have the genes doesn't necessarily mean that I'm going to express the genes. And that's where all of the other regulatory factors that we have been discussing all semester come into play. What are the feedback mechanisms turning on and turning off genetic regulators so as to allow for those genes to be expressed? Some of the regulations that take place due to metabolism can lead to what's referred to as epigenetic regulation. That is where metabolism has caused the genes themselves to be permanently turned on or permanently turned off. all cells that we have within our body come from the first zygotic cell. However, all cells do not exist in the same manner as that primary cell. And the reason for all the cells functioning the way the cells do is because cells have been epigenetically fixed through differentiation and then specialization within the fetal development sequences. There are secondary non-fetal sequences that can do the same type of epigenetic regulation, in which we can express or unexpress traits through methylation and demethylation of histones and regulatory proteins. Or we have secondary genes that will lead to an enhancement of expression, something that we see with a gene like the agouti gene, where we won't actually express distinct phenotypes without that agouti gene also being expressed. So let's take a look at how we can use these Mendelian laws so as to look at genotype and phenotype, and we'll look at a mono and then a digenic crossing. So we'll start with a mom and a dad genotype of big A, little a. So we have a dominant, the capital, and then a recessive, the lowercase. Now, in most complete dominant cases, if we have a dominant allele there, it's going to be expressed. If we have two copies of the recessive allele or the homozygote alleles, then we're going to express the recessive, in which we can set up a probability table referred to as the Punnett square, in which each possible allele from each parent gets laid out in its own row and its own column, mom on top, dad on the side. And we simply just fill in the table where the dominant always goes first and the recessive always goes second. And so in this case here, we get a ratio of a 1 to 2 to 1 ratio in terms of genotype, or a 3 to 1 ratio in terms of dominant expression versus recessive expression. We can take this and actually look at a slightly more advanced Punnett square, where we are now looking at a diallelic crossing. And once again, we simply just fill it out. And so what are the possible outcomes? Well, we can either get the big A and big B combined together, or we get the big A and the little b combined together, or we can get the little a and the big B combined together, or we can get the little a and the little b combined together. And then once again, just like we did before, we simply fill in all of the possible combinations based off of that interplay. And so what we end up getting is we end up getting distinct ratios of the expression of the dominant A and the dominant B, the dominant A and the recessive B, the recessive A and the dominant B, or the all recessive. So how do we go about getting that separation, the ability to get the A's separated from the B's and the A's separated from each other? It starts with the increase of genetic diversity issues in the process of crossing over. And the crossing over is the ability to exchange genetic loci, genotypes, the genes, from homologous chromosomes, establishing in what's referred to as analogous chromatids within the meiotic process. And so what's up happening is that we have junction points, and those junction points lead to the production of the analogous chromatids following prophase 1 within the meiotic process. So if we look at the meiotic process, we're going to have back-to-back cell divisions without having a growth phase in between. And so we don't get a secondary replication of genetic material. We break this up into what's referred to as phase 1 and phase 2, in which we get the interphase leading to the synthesis phase, leading to the growth phase 2, going into prophase 1. Prophase 1 will lead into metaphase 1, and metaphase 1 will lead into anaphase 1, and anaphase 1 will lead into telophase 1, which will lead into cytokinesis and the formation of the secondary gametes. We then get immediately into prophase 2, which leads to metaphase 2, which leads to anaphase 2, which leads to telophase 2. And notice that each one of the cells coming out of meiosis 1 will undergo a meiosis 2. And so at the end of meiosis, we get 4 cells that have half the complement of protein, excuse me, half the complement of genetic material from the initial cell that entered prophase 1. The way in which the cells go about doing this is going to be differentiational based off of whether it's a spermatogenic process or an oogenic process. And so let's take a look at the spermatogenic process. And so the spermatogenic process starts with the spermatogona going forming from the stem cell through normal mitotic division. We then get differentiation into the primary spermatocyte. This primary spermatocyte will then undergo secondary divisions, leading to meiosis 1, forming the secondary spermatocyte, going through meiosis 2, forming the primary spermatid. The spermatid will then undergo what's referred to as the spermatogenesis, and the spermatogenesis will form the spermatozoa, and the spermatozoa will form into the sperm. As it goes through these processes, the amount of organelles contained within the dividing gamete becomes lessened, and we get specialization of the remaining organelles within the spermatogenesis process. The entire process takes place over about a 65-day period from the initial division from the stem cell to the terminus formation of the sperm. Each day, there are about 300 million sperm that are completing spermatogenesis across both testicles within the male. Upon finalization, there's about a 48-hour window of viability for the sperm, whether it is contained within the epididymis or ejaculated into the reproductive tract for the female. So that is spermatogenesis. Let's take a look and differentiate this from the oogenesis. And so when we look at oogenesis, we have a differential place to start with. It begins during fetal development, in which the oogonium will develop into the primary oocyte. This division from oogonium into primary oocyte gets truncated within the first meiotic phase. And so what ends up happening is that the developing ovulate is only going to come from these primordial ovulates containing the primary oocyte. All of the rest of the potential cells within the ovary will develop into the secretory cells associated with the ovulate and with the endocrine functions that we look at. Following stimulation of the ovulate from FSH and from estrogen, and then from FSH, LH, and estrogen, that primary oocyte will continue through the final terminus of meiosis 1, forming the secondary oocyte. This is still a 2N cell. This 2N cell will then be ovulated within the ovulate. This ovulate will interact with the sperm, leading to the initiations of fertilization. It is during this phase that a rapid secondary meiotic division takes place. That rapid secondary meiotic division ensures that a majority of organelles will remain within the secondary oocyte and not be found within the discarded polar body. That leaves to the ovum, which will fuse with the sperm to form the zygote. So notice we have two distinct differences in terms of the process of oogenesis, in terms of its start and its terminus, as well as the total number of oocytes and ovulates relative to the male. Even though we talk about this leading to increase of genetic diversity, there are issues that can come about. In the study packet, I've given you a list of various problems that can come about. These include genetic abnormalities. Genetic abnormalities typically stem from either meiotic issues, such as non-disjunction, leading to trisomies, unisomies, or nullsomies. These tend to be seen more within females than within male in terms of the gametes, which means that we see more of these meiotic issues within the ovulates as opposed to within the sperm. And please try to think about why that might be. We will also see mutations taking place, and we'll see epigenetic changes taking place. We used to think that the epigenetic changes that took place during one's lifespan got wiped away from the gametes, but what we've been finding out recently is that epigenetic changes that took place within your lifespan are being fixed within your gametes and are being seen not in your children's, but in your grandchildren's expressions. We will see structural abnormalities. The structural abnormalities tend to be due to protein denaturing, which we'll see more often in males than we will see in females. And once again, try to think about why that might be. We will also see organelle issues. This organelle issue is going to impact with the female gamete production as opposed to the male gamete production. So let's take a look at some of these abnormalities that might take place. So in terms of genetic abnormalities, there are a couple areas that can occur. We can have points of non-disjunction in both meiosis 1 and in meiosis 2. The points of non-disjunction will lead to the trisomies, the unisomies, and the nullsomies. We can have issues with methylations and with mutations that take place during the normal interphase development within the growth phases leading to the primary reproductive gametes. So why do some of these issues come about? Well, there's issues such as the age of the gametes in which they become less responsive to the actual signals coming about. There could be issues, particularly with spermatogenesis, due to heat and restriction of blood flow. Remember, these are aerobic obligated tissues which need constant rates of perfusion in order to normally do their metabolism. We can have lifestyle effects such as diet and physical activity issues coming about, causing changes within gametogenic processes. We can have certain types of teratogen exposures, which include chemical exposures from mutagens and carcinogens, leading to issues within the gametogenic process. We can have exposure to environmental toxins, which have both latent and immediate effects. Some of these environmental toxins will act as endocrine disrupting chemicals, and I've given you a small little reading material on endocrine disrupting chemicals associated with the reproductive physiology. There's exogenous hormone abuse, in particular androgen abuse as well as estrogen abuse. And then there are infections, in particular STD infections, that can lead to some of the abnormalities. So let's take a look at what these abnormalities will come about. And so about 21% of all genetic abnormalities will come strictly from the oocytes. About 9% of genetic abnormalities will come strictly from spermatocytes. Of these, we get the predominance of issues coming in chromosomes 21 and 22, chromosome 16, and chromosome 23. Structurally, we're principally going to be seeing these within the sperm, and these are linked to cytoskeletal protein denaturing and changes within the cleavage furrow during the meiotic process, in which we will have distinct types of sperm that are abnormal. And once the sperm becomes abnormal in terms of its structure, its ability to become motile and move diminishes, leading to its reduced viability. And it's one of the reasons why we see more issues in terms of gamete functions within the sperm as it relates to structure. There's very rare abnormalities in terms of structural issues with the oocyte. Most of these are associated with cytoplasmic and organelle issues, and we'll talk a little bit about that when we look at the fertilization stages leading to the formation of the zygote.